Good evening.
Good evening, sir.
Okay. Yeah, good evening. Please unmute your phone. Okay, so now in this session, one of our friend, senior friend and academician, good academician, scientist, professor from Biotechnology department by name called Dr. Ramachandra Kini is going to deliver a talk on current and emerging DNA sequencing technology. Current and emerging DNA sequencing technology. He is expertise in the molecular biotechnological aspects. He is from Biotechnology department, University of Mysore. Biotechnology department is one of the best department among all the science departments in Mysore University campus. So sir has graduated, post-graduated and obtained his PhD from Mysore University only. And Dr. Ramachandra Kini sir, area of specialization are many. Just to quote few: plant molecular biology, molecular plant microbe interactions, genetic profiling of medicinal plants and their endophytes. So sir has been awarded and recognized after his research experience. So as a senior research fellowship award from CSIR, Government of India, Young Scientist Award in 86th Indian Science Congress held at Anna University, Chennai. Guest Scientist Fellowship from Danida, Denmark. Biotechnology department is known for Danida projects, and he has been awarded Guest Scientist Fellowship from Danida. Then Boys Caste Fellowship from Department of Science and Technology, Government of India. And for this, he had been to Netherlands for some time. Then he has been awarded Professor H.C. Dubey Outstanding Young Scientist Award in 2012, instituted by Indian Society of Mycology and Plant Pathology. Of course, sir has brought a lot of funds to his department and funding agency mainly from DST, DBT, UGC, MHRD, UGC and UGC. So worth more than 2 crore rupees funds he obtained from different funding agencies, Government of India. Sir has guided more than 9 PhD students successfully, and currently more than 6 students are working under his guidance. And 6 students have obtained MPhil degree. Sir has published more than 85 research papers in reputed foreign journals with very good impact factors. He has attended so many conferences as well as organized conferences and presented research findings on various platforms at the national and international level. Of course, sir is member to various organizations. Just to quote a few: Indian Science Congress Association, Indian Society of Mycology and Plant Pathology, Society of Biological Chemists, Indian Phytopathological Society, Association of Microbiologists, and Indian Botanical Society. With this brief introduction, I welcome on behalf of MHRD UGC Academic Staff College, I welcome you, sir, on behalf of Department of Geology, University of Mysore, to this session. Over to you, sir. Thank you.
Thank you, Mr. Basaraj, for the nice introduction. So it's my pleasure. Good afternoon, everybody.
Is it audible?
Good afternoon, sir.
Good afternoon, sir.
Good afternoon, sir. Yes, sir.
Good afternoon, sir.
Yeah. So it's my pleasure to be here to interact with the young faculty across the country. And my lecture topic, as the professor has already mentioned, current and emerging DNA sequencing technologies. I'm sure you're all familiar with the central dogma of molecular biology, right? Which was actually proposed way back in 1958 by Francis Crick, according to which the DNA is the main component molecule which actually holds the genetic information, and it replicates itself it makes copies of itself by replication. Then DNA is converted to RNA by transcription, and RNA gives rise to protein, which are all the workhorse. So DNA is the main molecule which actually holds the genetic material. So our main interest today is this molecule, DNA. So this sequencing of the DNA. So it's the process to determine the order in which these bases are present along the DNA strand. And in the past decade, this sequencing, nucleic acid sequencing, has increased exponentially. There's a great rapid pace by which the sequencing technologies have developed. And now the sequencing technique has become accessible to routine research and clinical labs all over the world. Now, the first major foray into this sequencing came from the in the form of Human Genome Project. I'm sure you're all familiar with that. It started in 1990 October, and it took almost 13 years to get completed. And in 2003, we got around 2000, we got the first drafts and almost completed in 2003. And this Human Genome Project, it was accomplished with what we now call as first generation sequencing, called the Sanger sequencing method, which was developed in around 1975 by Edward Sanger, and it is still actually considered the gold standard for nucleic acid sequencing. And still, after almost 2.5 decades, it's still the gold standard for Sanger for the sequencing. Now, this indicates the different milestones of the sequencing development throughout the history of molecular biology. And the Sanger sequencing and the another sequencing which came along with that is what we call as the Maxim and Gilbert sequencing at around 1977. They are now what we call as first generation sequencing. Then after that came around 2000 plus second generation sequencing, which we call next generation sequencing. And the major technologies were from what we call as pyrosequencing. And then you have the Illumina company coming up their own technology. Then ligation based sequencing in solid ABI, and ion torrent, the semiconductor based sequencing. Then after that, even more advanced technology came, which we call third generation sequencing, or the next generation sequencing. And there are two important technologies here. One we call as from the PacBio lab, sequel systems around 2011-12. Then you have the nanopore from the Oxford group. So this, what we now call as first generation, second generation, third generation sequencing technologies, there are different theories, principles on which they are based. Now this Sanger sequencing or the Maxim and Gilbert sequencing, they infer the identity of the nucleotide using the DNA base, and then there is separation by electrophoresis, gel-based electrophoresis. And the fragment length were from 500 to 1000 base pair in that. The next generation sequencing or the second generation, this is what we call as high throughput sequencing, also called as massive parallel sequencing. Parallel sequencing of several pieces of DNA together parallel. But the read lengths are relatively shorter, 50 to 500 base pair. Now, what it means is 50 to 500 base pair sequences you get, and these are sequenced multiple times to get the accuracy. So parallel sequencing from the Solexa, ion torrent, Illumina, etc. Then the third generation, which we call as long read sequencing. Here, the sequencing of the DNA is carried out in real time at single molecule resolution. So single molecule is sequenced, and very large size DNA is sequenced here. Tens of kilobase fragments will be there on an average. So little briefly about the earliest methods. So in 1977, actually, two separate methods came up, which one we call chain termination method or cycle sequencing developed by Sanger et al. And then chemical degradation method or chemical sequencing by Maxim and Gilbert. Now, to begin with, both were quite popular equally. But for some or the other reasons, the cycle sequencing method is still used. And somewhere down the line, Maxim and Gilbert lost its popularity. And as of now, nobody is doing a Maxim and Gilbert methods now. So this method is based on the principle that when you have a single-stranded DNA molecules, which have difference in length by just a single nucleotide, they can be separated using polyacrylamide gel electrophoresis. So we generate fragments. This is the basic principle of the Maxim and Gilbert sequencing. I'm sure you all are familiar with this. You start with a duplex DNA and label the ends, 5' end are labeled with a radio label, and then you cut with a restriction enzyme. then generate single-stranded samples, and you expose the samples to different chemical methods, reactions, which will break the DNA after specific bases. That is, G means after every G residue, it breaks the DNA on A plus G, then C plus T and C. So there are chemical reagents carried out, reactions carried out to break the DNA after that respective bases. And these four are the sequencing reactions which are separated on polyacrylamide gel. And since the ends are labeled with a radio label, it is then exposed to X-ray film. You do autoradiography, and then you read from the bottom to the top. That's a maximum Gill part. The Sanger's method is what we call as a dideoxy method or chain termination method, which uses a dideoxy NTP, which is a 2' 3' dideoxy. There's a lack of OH group at both the 3' and the 2' positions in the ribose. So because of which it terminates the chain when this dideoxy is incorporated. So we add this didein. So one DNP is radioactively labeled, and then you add this one of the DDNTP. So randomly, there are chances of the chain breaking after every residue. And each one is labeled with a different fluorescent dye. And then after the reaction, when you separate using capillary electrophoresis, followed by a laser detection, the fluorescent labels, it will come out like this. This is a chromatogram, and you read the bases of different colors. So this is still being carried out. This is capillary-based sequencing. And there are automated DNA sequencers like 8 capillary machine, 360 capillary machines like that. So you can do large number of samples at a time. So this was the method which was used for the large-scale DNA sequencing of the Human Genome Project. This is one of the typical labs in US where they did the large-scale DNA sequencing. Every day, around 120,000 DNA sequences were generated from around 60,000 plus plasmid preps. So what's wrong with this Sanger sequencing, even though we call it as a gold standard? Now, these methods is good only for small reads, what we call 500 to 750 base pair. And they are relatively expensive, and it takes some time because there is a gel electrophoresis. You have to, after reactions, you have to separate the components by gel electrophoresis. And human genome is about 3 billion base pairs. That's why it took so long, based using only this Sanger's method. It takes very long time for complete sequencing of the large genomes like human genomes. So the next generation sequencing came because of the increased demand for cheap and faster methods of sequencing. So that's how we call it next generation. Now what they do is they carry out massive parallel sequencing, multiple sequencing parallelly, during which you get millions of fragments of DNA from the single sample. And because of this, it can be high throughput sequencing, and it allows sequencing of the entire genome in less than a day. So it's much, much faster. So here the sequencing is by synthesis. Advantages is that it is accurate and there is parallel processing, and it can be automated easily. And there is no need for gel electrophoresis in this method. So these are some of the important next generation sequencing platform. And they started with several companies came up with their own ideas and machines of next generation sequencing. But currently, actually, only 2 are running in the market. So we started with the 454 sequencing. The Roche company, they came up with 2 systems. And then you have the Illumina method, which is still in use these days. Then the applied biosystem, the solid life technologies, they come with the solid systems, 2 systems. And then the Ion Torrent Life Technologies again came up with another semiconductor-based sequencing technology. So these are the next generation or the second generation sequencing. Then the third generation, where you have this Helicos generic analysis system, then the PacBio or the Pacific Bio systems, and the Oxford Nanopore. 3 systems started the third generation, but currently we have only 2, the Pacific Bio and the Oxford Nanotechnology. So we'll be discussing the principles of these technologies. So a typical NGS or the next generation or second generation sequencing workflow starts with the preparation of the library. So you isolate the DNA and you prepare the library. Then you carry out either emulsion PCR or Polyny PCR, depending on what type of technologies you are following. Then after emulsion PCR, you can do semiconductor sequencing, which is the basis for the Ion Torrent machine, or pyrosequencing, which is the basis for the Roche system. or you can do sequencing by ligation for the solid. The Polyny is colony on a plate. Polyny PCR on a plate. It's called Polyny PCR on a slide. And this is the basis for actually the reversible terminator sequencing technology of the Illumina. So we'll be mainly looking at the ion torrent Illumina and little bit about the pyro sequencing methods. The solid sequencing is obsolete now. Nobody is doing that. So we'll not go to that in detail. So library preparation, it starts with isolating the DNA. Then you fragment the DNA by either enzymatic digestion or sonication. You get random fragments. Then you attach the primer and the adapter on either side of the fragment. So this is a typical good fragment. This is the center. You have the sequence, and then you have an adapter sequence and a primer sequence. Then you select the size, select the fragments. Then it is subjected to your emulsion PCR after library preparation. So in the emulsion PCR, you are carrying out the PCR in a small tube in an emulsion. So this emulsion contains emulsion oil, lipid, and then you have the PCR mixture and a small bead and the library DNA. And this is by mixing, you get it in emulsion. So this emulsion, you will have multiple thousands of micro beads, micelles like. So everything, each one is encapsulated, a small bead. This is called a microreactor, ideal microreactor. Typically, it contains one strand of the library DNA, the primers, and one bead, and the PCR mixture. So when you start the PCR in this emulsion, there is first denaturation of the library fragment. The two strands get separated. And when you do the annealing, you know the typical PCR reactions. After denaturation, you do the annealing. One of the strand, that is the reverse strand, it gets annealed to the bead because there are sequences, pieces of single-stranded DNA attached, and that will be complementary to this. So it gets, the reverse strand gets attached to the bead. In the third step, the extension, the polymerase will amplify the forward strand. Okay, starting from the bead towards the primer side underside. Then again there is denaturation. These two strands will denature from the bead, and the forward strand is connected to the bead by the sugar phosphate, because that is the newly synthesized strand during the extension. So now that is covalently attached to the bead. Then again there is annealing. Now these primers, what is added, it gets annealed onto the single-stranded forward strand which is attached. And then when you do and that's what happens, then you do the extension, the polymerase will now extend from the opposite strand, from the primer towards the bead. And this you repeat multiple times, and you will get in each microreactor a bead with multiple double strands attached to the bead, each piece of DNA. Like this, there are thousands of pieces in this emulsion, and almost entire the DNA content, it is actually amplified in that emulsion PCR. On the Polyny PCR, here you are doing what we call as bridge amplification. This is the basis for the Illumina technology. So here we prepare the genomic DNA samples, randomly fragment the genomic DNA, and then you ligate adapters on both the sides of the DNA. Then this is added to what is called as a flow cell, which contains a glass plate, the flow cell channel. This is attached, the DNA is to the surface, and it will bind the single-stranded fragments randomly inside the surface of the flow cell. Then we do amplification, add the unlabeled NTPs and the enzyme polymerase to initiate the solid phase bridge amplification. So there is a binding of the single-stranded DNA to this, and it starts extending the primers. It starts extending, and you will get a double strand. So fragments will become double strand after one amplification. So each one you have an attached, one side is attached, another strand is free. On other side that is attached, this is free. So this forms what is called a bridge. That's why it's called the bridge amplification. So then you denature, it becomes single strand, and then you repeat the cycle. Upon multiple cycles, you have an array of single molecules. Okay? Several million dense clusters of DNA are generated in each channel of the flow cell. So this is the second step, that is emulsion PCR or Polyny PCR. Next, the technologies, you have semiconductor and pyrosequencing. then the Illumina technology. So first we look at the pyrosequencing technology, 454 machine from Roche Biosystems. So the workflow involves library preparation, emulsion PCR, and then pyrosequencing. Now this pyrosequence, the basic idea here is that the visible light is generated, and the light generated is proportional to the number of nucleotides incorporated. So nucleotide incorporated by DNA polymerase is coupled to light generation by luciferase. So this is the overall reaction, 3 reactions occurring. There are 3 enzymes put together. So the template DNA plus incoming nucleotide by polymerase, it will generate a pyrophosphate. I'm sure you are all familiar with the DNA polymerase reaction. There is a generation of pyrophosphate after every reaction. Now this pyrophosphate is made to react with this adenine phosphosulfate, where there is a adenine and you have the phosphate and the sulfate group. So the enzyme ATP sulfurylase will convert this pyrophosphate and APS to ATP and sulfate. Now this ATP in the third step, this ATP is made to react with luciferin in presence of oxygen by the enzyme luciferase, and it will emit light. So the light here emitted is directly proportional to the nucleotide addition. Okay. So 1 picomole of DNA will generate this much of ATP and 6 into 10 power of 9 photons at 560 nanometer. So what is done? You immobilize the DNA, 3 enzymes are added, then there is a wash step to remove nucleotides after each addition. So every addition, whenever there is a incorporation of a nucleotide, there is emission of light, and that light is captured. So light is captured means that the added NTP is incorporated. So there is sequential addition. So what we do is we add the beads in the picotiter plates, around 1.6 million wells in a plate. So we load the beads and the emulsion PCR beads. This is how it looks. And then we add the reagents. So there are 4 nucleotides are added sequentially, one after another. So you have the DATP, after that CTP, GTP, TTP. So every time a NTP is added, if it is incorporated into the growing chain, it emits light. If it does not, it gets incorporated, there is no light. So we know what you added. When ATP is added, if light comes in, that means that sequence, that particular position is A. When C is added, there is light, that position is C. That's how it is read. Otherwise, it gets washed off. So you have the luciferase, it will give out light, and that's how you read, depending on. So this is the nucleotide addition order, and whenever there is light, that is taken as that's how it is counted. There is A, C, G, T, like that. That's the pyrosequencing technology. The second is the Illumina technology. They have various machines like HiSeq, HiScan, Geome Analyzer, MySeq, like that. And the basic workflow is you have the library preparation, there is bridge amplification, and then there is the technology here is reverse termination sequencing. And the various machines, they have got various capabilities, and the read lengths can be as low as from 100 base pair to up to around 250 base pair. And throughput can be very high, up to 600 GBs can be sequenced in a run, and 3000 million reads can be carried out in a run. And the runtime accuracy is quite high, 99.9% accurate. So this is the sequencing pipeline for the Illumina, where you prepare the samples around 1 to 5 days, ligating. Then you do the bridge amplification in the flow cell. This is how a typical flow cell will look like. And then you do the sequencing and imaging. This mainly involves capturing the color images. And then you do the analysis. So how it is done is you have this what is called a reversible terminator sequencing chemistry. So we add, so this is the bridge amplified component, single stranded there, and there's a primer attached to that. So you add the sequencing reagent, the first base gets incorporated, the unincorporated bases are removed, then you detect the signal. Then next cycle. So every time you are adding, this is a typical nucleotide which is added. There is on the 3' OH, there is a block. 5' there is 3-phosphate, and there is a block on the 3'. So that it doesn't, unless you remove that, next step cannot happen. And there is on the base, there is a fluorescence dye attached. For each of the bases, one colored dye is attached. So if A gets attached, there is one color emitted. If T is attached, another color is emitted. So that color is captured by the imaging system. So after it is added, you wash off the unincorporated bases, you detect the signal, then you add the deblocking. This floor is removed and this block is removed. Now it is ready for the next. So 3' OH becomes free, the floor is removed, the die, and then it goes to the next cycle. So this is repeated multiple times. These are typical steps. In the left side, top left side, here you add all 4 nucleotides together. Now each one will have a label with a different dye, different color dyes there in that. And this is on the plate, a typical bridge clonal array of the DNA which has to be sequenced. So there will be a primer on one side and you will add. So depending on the base there, the incoming NTP, it gets attached. So 4 different bases, they go and attached to their respective positions wherever there is the complementary base area. So 4 bases are attached. We wash, remove the unincorporated one, then 4 color imaging. So each one is a different color, we image, and this is how you get on the panel their colors. And then you cleave the dye and the terminating group, wash it, and then again go for the next cycle, next set, then again wash, then again read. And this is read on a plate. It's a typical plate on a glass plate. There's a CCD camera which will capture the image at the entire plate, and every position is actually will be changing. For example, from the top, the left first box here, it is showing green color, which means C. Next, on top of it, you get the blue, which is A. Next, you got red, this position here, which is T. And then again, you have another green, that is C. Then again, you have yellow, which is G. So that is how every position, it is read by the computer system, image analysis. So each dot, actually, colored dot, indicates a particular position on the plate. This is under the microscope, it is the image analysis is carried out. This is what is called base calling from the images. So at this position, the 2 positions are shown here. First position, first base is T, second base is G, third base is T, like that. At that particular position, the color keeps changing because you are adding different dyes. So every position, every dot, it is actually read by the system, and you will get. So this is parallelly. Now this piece and this piece may be same to some regions. So parallelly it is sequenced multiple times. So identity of each base in the cluster array read from the sequence of images. That's how you get the sequencing. Next is the semiconductor sequencing technology, or the ion torrent from Life Technologies. This workflow involves library preparation, emulsion PCR, and then sequencing using semiconductor sequencing technologies. So there are 2 important machines from ion torrent. One is PGM, called personal genome machine. Then you have the higher version called ion proton sequencer. In the ion proton, the throughput is much higher, up to 100 GB they claim, and even the reads per run is very high, and accuracy is close to around 99%. So how do they make? So this is based on the chip sequencing. It's also called chip sequencing. So very thin, ultra thin wafers are cut from the silicon bowl, and the transistors and circuits are then pattern transferred onto this, and they are etched onto the wafers using this technology called photolithography. And this process is repeated multiple times to get multi-layer of very thin, ultra thin circuits. So it basically is a simple chemistry and very rapid and fast detection. So this is how a typical ion semiconductor chip will look like. There are multiple layers. So there are microwells on top on a semiconductor chip where you are putting that beads from the emulsion PCR. Below which you have this. So there's the ion sensitive layer. Below which there's an ISFET, that is ion sensitive field effect transistor. There's a sensor layer will be there. And each micro well here, that will contain one single standard DNA template DNA molecule to be sequenced and one DNA polymerase. So there are different generation of chips, first generation, second generation, third generation, depending on how many sensors are there. In the first generation, there are around 7 million sensors. So that means 7 million pieces of DNA can be parallelly sequenced. Likewise, in third generation, you have 12 million sensors present there. So this is the basic chemistry of the semiconductor sequencing. Now, whenever there is a nucleotide incorporated in a polymerase reaction, a hydrogen ion is released as a byproduct. This is how it is. Okay, so you have this DNDP going out, incoming DNP when joins there, that OH from the alpha phosphate, it gets released, the hydrogen ion. So what this system, a semiconductor system does is basically is measuring this proton which is generated, released. Okay. So, and this gives very fast and rapid detection. So nucleotides are sequentially flown. So sequential addition, one at a time, A first, G, A, T, G, C, like that. So if the complementary base is there, upon there is incorporation, the nucleotide releases a single hydrogen ion, which creates the change. There is a pH change because of the hydrogen ion. And this is sensed by the sensing layer, which binds to this hydrogen. And it is transmits this to this field effective transistor gate. And the gate register a voltage change between the source and the drain, and which is proportional to the number of bases incorporated. So basically, this is a very elaborate, very expensive pH meter. You are just measuring the change in pH because of the production of this hydrogen, and very, very sensitive one. So this is how it is done. You have 4 nucleotides which are added sequentially, one at a time. And if nucleotide is incorporated, then hydrogen ion is released. And that is measured by the sensor. So there is no camera, just it's a pH sensor. So when you add a T, if it is incorporated, you will get a change. So that is what is measured. When nucleotides don't match, no hydrogen released. So there is a dash. So likewise here. And then there are 2 hydrogens are released. You get the intensity of the peak will be high. So from based on this is the average corrected ionogram from where you would read, depending on which base when you add, there is a pH change and how much pH change. So when you say long for the T here, peak here, that means there are around 3 bases. So there is 3 continuous, 3 or this is 4 up to 4 continuous T's like that. So that is how it is read. So basically it is based on measuring the change in the pH of the ions. So these are the 2 important NGS platforms which are currently under operation. The Roche machine is no longer used these days. So Illumina and the IF technologies. So you have mini-sec, MySec, then you have the next-sec, high-sec X like that, depending on the output. Okay, the high-sec X can read up to around 6 billion reads, up to 1,800 gigabase output can be. So for the production scale, human whole genome sequencing can be carried out by this, and this can happen within 2 to 3 days. Unlike the first HGP, which took almost 13 years, now you can sequence the genome by at least in around 2 to 3 days. Then the PGM and the proton, the semiconductor-based systems, up to 400 base pair length, and they can go up to around 10 GB. They are relatively lesser than Illumina. So currently, in the NGS platform, the Illumina is the market leader. Most of the sequencing, next generation sequencing, is being carried out using Illumina platforms. So the third component is the 3rd generation sequencing. Now this is what we call as real-time sequencing of single DNA molecules, or even RNA can be sequenced with this. So here there are 2 strategies used. One is sequence by synthesis. Here the nucleotide is incorporated and signal detection, they occur continuously in real time, as fast as the polymerase is incorporating the nucleotides. The average rate at which DNA polymerase works is approximately 750 nucleotides per second. So this is synthesizing at this rate, and you are detecting at almost at the same rate, 750 NTS per second. And this is what the technology used by SMRT, single molecule real-time sequencing, and that's the basis for the Pacific Biosystems machine. Then you have another strategy without synthesis or ligation, which is from the Oxford Nanopore. And both these technologies will give very long read, 1 kilobase, 1000 bases to around 2 megabase length. DNA can be sequenced in one go. So this is the comparison of the short read versus long read lengths. So short read lengths are the ones which come up from the next generation or the 2nd generation sequencing, where you have multiple pieces of small short reads they have to be computed, they have to be put in analysis, and the overlapping sequence you have to read, and then you will get the final. So the long read will have got advantage because they simplify the assembly process and provide much higher quality alignments, and they generate the finished sequence much faster and more accurately than the short read sequences.
Sir,
Yes.
how reliable is the long read sequence, sir, or how accurate it is when compared to short
accuracy,
reads?
because the third generation sequencing, as of now, it is relatively less accurate, but they are developing the technology towards more accuracy. But compared to NGS, currently the accuracy rate is little bit higher than the short read ones. But if you want to increase the accuracy, you have to do it more multiple times sequence parallel. So for example, one piece, this piece, if it's sequenced 4 or 5 times, the accuracy is better than when it is sequenced 1 or 2 times. So 10x times you do it, the accuracy increases. So you need more output, multiple times. It has a particular piece of DNA has to be sequenced multiple times. More number of times that is sequenced, the accuracy increases. Because out of 10 times you sequence, you just compare all those 10 sequences. And the one which is most consensus, that is taken. So accuracy increases when you have that is what actually happening in short reads. When you're having short reads, it is massive parallel sequencing. Each piece of DNA is sequenced around 20-30 times. So that's how the accuracy increases. So if that is also done in the long read ones, then the accuracy will be comparable to what you get in the short reads. Okay, this SMRT sequencing. This is the basic principle behind this. So it has got a very small nano scale level reaction cell. And each cell contains one DNA polymerase, which is immobilized on the bottom of the reaction cell. And this reaction cell is called ZMW, zero mode waveguide. Now, this cell at the bottom, it is immobilized by a modified 529. There is a bacteriophage 529, and a DNA polymerase is isolated from that. It is modified from that recombinantly. And this is a single subunit enzyme that is immobilized at the bottom of this ZMW microcell. And each sequencing plate contains thousands of these individual cells. Each cell will have only one DNA molecule. So each cell you are doing one DNA piece sequencing. Like this, you are doing parallelly for millions of pieces. So what is the basic chemistry here? You used a phosphate-linked DNTP, each of which contains one different fluorescence. There are 4 DNTPs, 4 different colored fluors. So during sequence, a single labeled DNTP enters the polymerase, and the DNTP, if it is matching the template, it is held in place. So upon reaction, the fluorescence signal is emitted because it releases the fluor in the ZMW for a very, very short period of time, nanoseconds. Then the DNTP leaves and the new DNTP enters. Okay. So this SMRT sequencing, here you are using the labeled phospholinked nucleotide. That is, the fluorescence is labeled to the phosphate, alpha, beta, gamma phosphate. In the case of earlier fluor addition, in the case of Illumina and all, the fluorescence is attached to the base. So it is base linked. Here it is not base linked, it is phosphate linked. So what happens is every time this DNTP gets incorporated, because the link between alpha and beta is removed, this fluor, it comes off. It comes off. So the fluorescent dye, since it is attached to the phosphate chain rather than the base, due to the natural synthesis process, the phosphate chain is cleaved when this nucleotide gets incorporated. So upon incorporation, this new phospholinked nucleotide, the polymerase will cleave this, and this will immediately diffuse. It will give out light, which is measured by this camera below, and then it diffuses away. It goes out from the background. Yeah, this is the detection and sequence determination. So fluorescence signal for each ZMW is collected, and data is collected as a movie of sequential signals. So you are adding all the 4 different fluors are there. So whichever fluor attaches, it will give that particular light, and that is measured. For example, you get light green color, you get it is read as A. Then next one comes in, then T, like that. So different colors will indicate different dyes, and hence you have different nucleotides. So sequential steps, fluorescent phospholink labels are introduced. Then the base being incorporated, held in this is the detection volume for tens of milliseconds, very, very small amount of time. And it produces a bright flash of light because it is illuminated from the bottom. And the phosphate chain is cleaved, it will release the dye molecule, and the whole process is repeated. This is the size of the chip. It contains thousands of defined nano well ZMWs, and each one is having a diameter of around 70 nanometers, and depth will be around 100 nanometers. And total detection volume in each of this well is around 20 zeptoliters. That is 2 into 10 to the power of minus 21 liters. So very, very, very small volumes. And this is the chip. You can hold it on your finger, and there's the size of the chip. And this is what it contains. Each chip contains millions of these wells. This is a typical scale of 43.5 micrometer into 32.8 micrometer tall. This is a multiplexed ZMWs, multiple wells. So DNA, this is the overview. The DNA polymerase is anchored to the bottom of the zero mode waveguide. So when you illuminate from the bottom, this laser light bends only up to around 30 nanometers. This length is around 100 nanometers. Laser light captures only bottom 30 nanometers. So when it illuminates, it is captured. Whatever color is light, that's emission is captured by the camera, and then it detects what color dye flash has given out, and that's how it reads. Yeah, this is how it is. You have the plate, you have the multiplexed zero mode waveguide chip. On the bottom, you have the camera which will detect this. Okay, so this is how these are multiple ZMWs. If individually it is captured, and parallelly there is reactions are occurring, millions of reactions are occurring, and parallelly it captures all, and it will send it to the detector, multiple ZMWs, whatever is read and is processed, and then you get the final data output. So there are systems, Pacific Bio, there are different systems. There are two important systems, what's called SQL system. Another is SQL 2. SQL is the earlier version. There are 1 million ZMWs in a cell. The SQL 2, it has got 8 million, 8 times higher number of cells. So reduced project time. This is a typical flow in the template preparation for the Pacific Bio system. So what we do is we fragment the DNA, determine the concentration, break it, damage the repaired region, repair the ends, then we attach the ligase, the adapters are ligated to that. Then you purify this, then there is a specific on the adapters, you have the complementary primers which are annealed, and then the polymerase is bound to that, and that is attached to the ZMWs. This is the typical reaction which occurs. A small video indicating how it happens. This is the chip with the multiple ZMWs containing tens of thousands of zero mode waveguides. Each cell at the bottom, this is one of the smallest light detection volume. So each ZMW is illuminated from below. So wavelength of light is too large to allow it to efficiently pass through. So it cannot completely go to the top, only at the bottom. So attenuated light from the excitation beam penetrates only the lower portion. So once the incorporation occurs, dye, it measures. After dye moves away, it cannot detect. This is a single molecule real-time sequencing. The polymerase attached to the DNA here, DNA template polymerase complex. The bottom, then the phospholinked nucleotides, they are introduced. And the reaction, when it's occurring there, each of the four, each one is labeled with a different colored dye, fluorophore. And the base is held in the detection volume, and when it is incorporated, light is emitted. And that is captured by the bottom camera there. Depending on what color is captured, it reads the bases. And this is occurring parallel in thousands of ZMWs, this makeup. So multiple parallel sequencing will be occurring throughout the chip. So in the 8 pack, you have the 8 chips in parallel. So you can load 12 loaded smart cells in the 8 packs. So parallelly, multiple sequencing is carried out. So what are the advantages and limitations of this pack bio systems? Main advantage is long read lengths, which allow for greater certainty when you are doing overlapping sequences. And there is better resolution of the repetitive region. When there is a lot of repetitive region, it is relatively difficult to overlap when you have in the short reads. In the long reads, it becomes relatively easier. And it can sequence DNA with unusually high GC content. Normally in the regular Sanger sequencing, and also when you have the NGS, DNA with high GC content is relatively poorly resolved by the short read sequencing technologies. So these are the advantages. Main limitations here is the lower throughput and error rate. When it started, the error rate of the PacBio was close to around 10 to 30%. And the cost per base was also relatively higher. But with the release of advanced SQL platforms and advances in the design of the flow cell, where they increased the ZMWs to over 1 million. They started with around 150,000. And then also improving the chemistry of the sequencing, the error rates now, they show 3%. And recently, this was in 2021, 3%. And recently, they are now claiming up to 1%, as low as 1% error rate. The other third generation sequencing technology is what we call nanopore sequencing, developed by the Oxford Nanopore Technologies, ONT company. And here, there is no synthesis occurring. Here you are threading the DNA, single-strand DNA through a micropore, nanopore actually. So when it is passing through the nanopore, it is read. So DNA can be sequenced by threading it through a microscopic pore on a membrane. And the bases are identified by the way each base affects the ion which is flowing through the pore from one side to the another side of the membrane. So what is done is you have the membrane, charged membrane, one side and another side will be having different charges. So there are 2 proteins. One protein will unzip the DNA helicase into 2 strands, like a helicase, a double-stranded DNA. 2 strands are separated. One strand is threaded through the pore. The other second strand, the second protein, it creates a pore in the membrane and holds an adapter molecule. So this single-stranded DNA is threaded, pushed through this nanopore. And when it is flowing, threading down, you have each of these bases. There will be different bases here. And there is a flow of current from one side to another. Now, this flow of ions through this pore, it creates a current. And each base here, it blocks the flow to a different degree. It alters the current. If there's nothing is flowing, there's a constant current. When bases pass through that, it changes the current. Now, this change in current, this flow, change in flow, it happens to different extent, different degree to different bases. Each base will alter the current a slightly different time. So you can measure, you can identify what base this is like this. Okay, depending on the nature of alteration of the current, you can identify the bases. Okay, so the adapter molecule keeps the base in place long enough for them to be identified electronically. So what it basically does is you're passing a single-stranded DNA molecule through a protein pore, and then you're detecting each base as and when it is passing through the pore in the real time. So this is one of the miniature version of this Oxford Nanopore, mini ion. Okay. So this company, Oxford Nanopore, became the first company to provide a commercially available nanopore sequencer, and this was in 1915, and it was available to community in 2015. And available in 2012 actually. Very small portable device. So here the protein nanopore is set in an electrically resistant. There's a membrane bilayer, and there's a constant ion moving. So you have a standard current. Okay. So an ionic current is passed through the nanopore by setting the voltage across this electrically resistant membrane. So there's a constant current passed. Now, if an analyte passes through the pore or near its aperture, this event will create what we call disruption in that current. So you can see here a molecule coming in, it disrupts this. There's a disruption in current. Now, this disruption depends on the characteristics of the molecule. So when there are 4 bases, each base will disrupt the current in a different way. So based on this, you can identify, it is possible for the system to distinguish the 4 different standard bases, A, T, G, C, and also if there are any modified bases, even that can be detected by the nature of alteration of the current here. So there's a porous transmembrane protein, cellular protein of a diameter approximately 1 nanometer is used. Generally, there is what is used is a modified alpha hemolysin or Mycobacterium smegmatis porine A protein, which creates a pore. And the nanopore is immersed in a conducting fluid, which is synthetic bilayer, and there's a potential applied across the membrane. And because of the conduction of ion through the nanopore, an electric current will be there. And any base passing through it, it disrupts. So you see the top one, helicase, it opens up the strand and one is pushed through that. And as and when it is passing, the disruption in current happens, and that is read by the system. This is the sequencing workflow. You prepare the library and then repair the ends and add a DA tail. You ligate the adapters, and then that is threaded. This is how your molecule, when you load it in the flow cell, this is the flow cell from the top view. Each one will have one particular nanopore. This is the cross section of that, voltage applied, current flowing. And as the single stranded DNA is pushed through that, there is a change in current, and that is measured. This is the video showing how this happens. The Oxford Nanotechnology. This is the mini ion flow cell. There's a motor protein, there's an adapter sequence, and the DNA or RNA. Even RNA can be sequenced directly because there's no synthesis here. There's a nanopore through which single stranded region is they use this nanopore sequencing even in space. They carried out sequencing using this mini ion sequencing the DNA in space in the IS International Space Station. Yeah, these are the various versions of these ions. You have this mini ion, pitch ion, promethion, like that, using the different high throughputs. Okay, what are the advantages and limitations of this nanopore technology? The mini ion, the smallest one, which is highly portable, and they have shown that this is capable of sequencing even when you plug it to a laptop. So it's highly portable. You can take it anywhere in the field, and this has been widely used in 2015 when there was an Ebola outbreak, and it was used to carry out sequencing in the field. Okay? And it is also used to conduct first sequencing experiments in space. And there is label-free sequence determination of native DNA and RNA as well, because you are not using any polymerases. Like it is not DNA sequencing by synthesis. So just DNA or RNA threading to a pore. So without need for any amplification, you can sequence the DNA or RNA. And it can produce extremely long read lengths. But again, the problem is higher error rates. So much higher error rates, around 15%. But still, this came up in around 2015. But now currently they have reduced the error rate, and in 2022 something they are claiming that the error rate is as low as around 1%. So this is the comparison between these two important third generation sequencing platform, PacBio and Oxford nanopore technologies. And PacBio, you have the RS2, then you have the sequel systems, sequel 2, like that. And this is the average read length around 10 to 4 kilobases. Whereas compared to that, the ION, the Oxford nanopore technology, they claim up to 4 megabase read length, much higher read lengths. But the error rate around 10 to around 13%, which now currently they claim it has come down to around 1%. Runtime is less than around 30 within the 1 day for PacBio systems, and from 1 minute to around 700, depending on the size of the DNA, you can complete the synthesis in less than a minute or up to around 72 hours. And that the throughput what you can get, 14 TB maximum, terabyte size DNA, very large size DNA can be sequenced. So the applications of these third generation sequencing platform, and which platform of sequencing is useful depending on the research interest. So for example, for DNA sequencing, for whole genome sequencing, for whole exome sequencing, targeted sequencing, for transcriptome sequencing, these are the various options available. For de novo genome assembly, you can use either PacBio or ONT. But for mutational analysis, the NGS is still preferred because accuracy rate is relatively higher compared to the third generation platform. So there are several technologies where still people go for the next generation sequence, the Illumina platform for detection of small indels, detection of SNP variations. So after the SNP detections in NGS, finally they go back to the Sanger sequencing to confirm. So validation is always by going back to the Sanger sequencing, the gold standard. Like mutation analysis, you still have the NGS. Okay. For RNA sequencing, you have currently only the ONT platforms, because the PacBio, it is based on synthesis options where RNA polymerase cannot be used. So the nanopore technologies, you can directly sequence the mRNA and the microRNA sequencing. All those applications can be carried out on the ONT platforms. So because of the advent of the third generation sequencing technologies, the cost per human genome, sequencing of the human genome has come down drastically. So when it started in 2001, it was around $100 million for sequencing the genome. Okay. But later it came down. So as per Moore law, it should have gone like that. But currently, after the third generation sequencing platforms have come in, the average rate of or cost per human genome has come down to less than $100 for a genome. Okay. And cost per raw megabase of DNA sequence has also come down drastically to very minute levels after the. So it is much cheaper, and the time also required is also very less. So you can get the entire genome sequence in less than a day, and a complete analysis and all within 3 days you can get the reports. So what are the other methods, future methods which are under development, which we call as fourth generation sequencing? So there are advances in the nanopore technologies being conducted where they are planning to use solid state nanopores, which are engineered from synthetic materials. Currently the nanopores are all proteinaceous molecules. So they want to generate synthetic materials which are more durable than the proteins and biomaterials. So such as the graphene or carbon nanotubes. And this development of this highly portable Smith Giant is under progress, where you can attach this to a smartphone. So you can carry your DNA sequencer in your pocket along with your mobile phone, the smartphone. It can be attached with an interface, Type C or like USB interface, and you can carry out sequencing along with your smartphone, powered by the smartphone. So very small and miniaturized version. Then there are also developments towards single cell DNA RNA sequencing. That is, nucleic acid isolated from individual cells. Each cell you can isolate and carry out. So very small amount, around 6 to 7 picogram of DNA will be sufficient. Then directly in the tissues, performing nucleic acid sequencing within the tissues, in situ DNA sequencing. That is also under progress. And coupling it with microscopy, scanning, tunneling electron microscopy, people are trying to do sequencing technologies. So in conclusion, so mainly about this third generation sequencing technology, even though this has been in existence only for a few years, it has already established a large scale. You have DNA and RNA sequencing applications. Okay, and this more development in this third generation and the fourth generation sequencing technologies may bring to the end the NGS platforms, because the main disadvantage of the TGS is mainly the accuracy rate. With more advancement in this accurate variant calling and also efficient identification of these mutations, the NGS may become obsolete, and third generation platforms may further develop. Then both the production of the high accuracy reads and introduction of more accurate bioinformatic tools, they are required for making this TGS as the future platforms for the sequencing. So there may be more enhanced versions of TGS, or even newer, brand new fourth generation sequencing technologies with more new or unprecedented characteristics may also come up in future. Yeah, so that's the end of my lecture on this. So in the market now, you have all different kinds of options available for personalized genome sequencing with different prices, and you can choose a researcher. A person can choose which technology to be used for sequencing his research sample. So any questions?
Sir, you mentioned about the different technologies under third generation sequencing. Which would be the choice of sequencing among the third generation sequencing?
Yes, sir. Third generation
sequencing.
Yeah, third generation sequencing. Currently, you have the PacBio and Oxford Nanopore. Okay, Oxford Nanopore, actually, they are saying it is relatively more cheaper. It costs approximately around complete library preparation, everything. It may come to around $200 to $250 per sample. Okay, so in India, there are only one firm for PacBio and there is one firm for Nanopore, Oxford Nanopore sequencing. And Oxford Nanopore sequencing is relatively more cheaper than the PacBio systems.
Thank you.
Hello, sir.
Yes.
Sir, this is Kavya from Central University of Tamil Nadu.
Okay.
Sir, you mentioned this nanopore sequencing, which requires to use a lipid bilayer kind of structure and a protein. So every time we are doing a sequencing reaction, do we have to change this membrane, or
yeah, that is a chip. Stable? That is chip. It is present in the flow cell. So you have to buy the flow cells.
Okay.
Okay. All so right. You buy the flow cells and then load your sample. So every time you are using the chip, all these they come with these disposable chips, and these chips have to be used fresh every time. You have to use a new chip.
Okay, sir.
Same like in the case of these ion torrent machines, where the DNA is a semiconductor based technology, it comes with the chips. So it is actually basically the chip which is costing more, the maintenance costs.
Yes, yes. So let's say if we want to use third generation sequencing for a simple thing like a plasmid sequencing or a
For
small
what?
PCR, plasmid sequencing or just a PCR fragment sequencing.
Yeah, it is not advisable. It is very expensive.
It's not. It's like too much of technology for too small.
See, next generation sequencing or from NGS onwards, NGS or TGS, both are for entire genome sequencing, not
Yes,
just gene sequencing. When you're talking about only gene sequence, one gene, one PCR fragment, better go for Sanger sequencing, where they will charge hardly around 200, 300 rupees per sample these days. You will give you 400, 500 base or up to 1000 base sequence. So when you have only one sample, one plasmid to be sequenced, it's better go for Sanger. When large amount of DNA, whole genome or whole exome, then we go for this NGS or TGS.
But actually, I was thinking like how we have a nano drop in every lab nowadays.
Right.
Maybe in future will we have like this nano?
Nano personal genome sequencers. That's what you can just attach it to the laptop, and because these machines, they are not very expensive. Okay, around $2,000, $3,000 you can get a mini ions machine. Consumables will cost later. Okay, but the machine cost is relatively lesser. So just like as you said, PCR machine or nanodrop, every lab which is doing large scale, every now and then, if you are doing the sequencing, exome sequencing, those kind of a thing, then people will have it will become as common as a PCR machine.
Yes, sir. I think that's the future soon.
Yeah.
Sir, thanks a lot for such a fascinating talk. Thank you.
Thank you.
Sir, the error caused by this nano is high when compared with the other one.
Yes.
So,
currently they are claiming error rate is only around 1%, 99% accurate. The recent reports from both these companies, PacBio and Nano, Oxford Nanopore, they're claiming now that the error rate is close to 99%. I mean the accuracy rate. So error rate is around 1%. But still, this is compared to your NGS, the error rates are still a little bit higher.
So according to you, a laboratory needs the third generation nano or the other one?
See, laboratory buying, if people are planning to go for the technologies, it depends on what kind of applications you are planning. See, if the if you are planning for, see, I put a table here like this. See, if you are going for let's say mutation analysis, third generation is not advisable because error rates are relatively higher, and mutation, it is just 1 or 2 base changes. So if you detect a mutation, you won't know whether it is an error in sequencing or an actual mutation. Likewise, SNPs, detection of SNPs is not advisable in third generation. You go for NGS and then validate it by Sanger sequencing. So depending on your requirement, research interest, we decide whether you go for NGS or for TGS. It's like that. For example, RNA sequencing, you cannot do by NGS at all now. You have only Oxford Nanopore technologies.
Thank you, sir.
Sir, if there are no questions, it's my pleasure to propose the vote of thanks on behalf of MHRD UGC Academic Staff College and my fellow participants. I wholeheartedly thank my all-time favorite teacher, Kini sir, for an excellent and dynamic talk on current and emerging DNA sequencing technologies. Thank you so much, sir. Thank you, one and all.
Thank you. Thank you all.
Thank you, sir.
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 Thank you, sir.